Yep, so I'm Tom, and I'm from Reken, and so we don't have any fun beavers, but we have a termite apparently as our mascot in Reken. So that there. And so the stuff I'm talking about today is a little less polished, I think, than maybe some of the really nice talks we've had, but hopefully it's a good basis for us to have some nice discussions today and going forward. Yeah. So we've seen lots of nice classifications from like Andy and Francisco, and yesterday from Manas. And so yeah, there's lots of different reasons that we might see conflict within individuals. Individual genes within the same organism might be able to grapple for kind of greater share of the reproductive output of those individuals. The same genomes in an individual might also have different valuations of the social partners. And also when they have strategies that span multiple types of individual, different genes within an individual might favor different balances between being good in one state and good in another. And that might also generate conflict. And so I think one of the things we're kind of looking at here or in this conference is maybe trying to see how examples of these conflicts fit into these categories, but maybe also are there examples of conflicts that maybe require a bit more thought about how they might fit into this. And so one aspect of this that I've been thinking about a little bit recently is kind of structure that exists within genomes. So, I mean, lots of the examples of conflict we've seen, often you think of just really a soup of genes in a single individual, but obviously that's not true. We know genomes are highly structured into kind of chromosomes and so on and so forth. And so we see lots of research at the moment ongoing about how we can leverage this kind of internal structure of genomes to make inferences about human history, for example, kind of ancestral recombination graphs and the like. But also lots of kind of new genetic work is finding examples of these supergenes where blocks of the genome are maybe going to have slightly different transmission patterns in kind of an ecological sense. And so we can think that if you are closely linked to some of these regions, maybe your transmission pathway through a life cycle might be different. For example, the nice example I have here is this. Can I do that? Anyway. The example I have here is of these fungus gnats, which are some of my favorite creatures. So they have this unusual split sex ratio system caused by this supergene on the X chromosome. And so depending on the combination of chromosomes you have, you'll be either a kind of exclusively daughter-producing or exclusively son-producing female. So we can imagine if you're kind of linked to this region, maybe the way you move through this pathway is going to be slightly different. So again, this sounds slightly unrelated, but so there's this paper that I read a few years ago. So actually it came out about 10 years ago. And I was really interested because it didn't obviously fit into some of the examples of conflict that we've been talking about, or at least not in a straightforward way. So this was a paper by Frederick Guillon. And it's kind of quite a simple dynamic, but what they have is they have an enhancer upstream of a gene, and there's some kind of mutation, selection balance ongoing at this gene. And then they consider this dynamic, whereas an enhancer, you can maybe draw down more gene expression onto your particular copy in cis. And they find this really unusual dynamic that stronger enhancers can be able to invade the population. So we see this kind of red lines are stronger enhancers, blue lines are weaker enhancers, and then we've got kind of recombination rate on the x-axis. And so what you can see is that if recombination between the enhancer and the gene is smaller, then you can get the invasion of these strong enhancers. While as you go further away, these enhancers the stronger enhancers don't necessarily invade. And moreover, they find that it's not really shown here, but you get this kind of long-term dynamic where these enhancers seem to compete to draw down more gene expression to their copy. And this can have some kind of transient deleterious effects for the organism, but also you think as well, maybe there's some kind of fixed costs where you're spending more energy on maybe gene expression, or it's kind of inappropriately allocated. So I thought this was really interesting because it didn't obviously, to me, when I was reading this, fit into some of the examples of the conflict we've been talking about, which is interesting. And actually, if you look at the citations to this paper, it's like 10 years old. There's lots of citations, but almost none of them are from kind of papers discussing intragenomic conflict. And it doesn't actually, lots of these recent reviews don't talk about this paper. So I thought that was interesting. It's like, do we understand, do we have a good intuition about what type of conflict this is and what's going on? So kind of what I wanted to talk about today is thinking about linkage within genomes and think about why, for example, in this example we've just looked at, why might there be conflict? And in particular, two aspects of this. First of all, how might different genes within the genome have different valuations? And that's one form of potential conflict. And the other is how different genes might be cause different distortions of the type of individuals they see in the population, and how this might also generate conflict. And at the end, I'll finish with some musings about maybe some of the are these conflicts more common than we might think. So the first example I'm going to think about is we have mutation selection balance. So I'm going to think about kind of a haploid sexual genome. So we've got kind of a region upstream in this yellow, and then we've got our mutation, good and bad, in the green and red. As you can see, that there's some kind of penalty, some kind of mortality rate maybe you pay if you've got this deleterious mutation. I want to imagine that there's some kind of mutation selection balance, so that the frequency of this deleterious mutation is Q, or approximately Q. So we can imagine that if you're this region upstream of this gene, there's various processes that might move you back and forth between these two backgrounds. There might be mutation at the locus, so there might be new deleterious mutations entered into the population. Alternatively, there might be recombinations. You might kind of, when you mate, you might be able to exchange onto the different genetic background. So one way we can think about this is rather than trying to explicitly map the LD or that we might do in kind of some pop gen models, we can instead maybe just think about actually, should we just treat these as two environments that we can move between? So our kind of focal gene can move between a kind of good environment, and it's going to have some reproduction in this good environment, and it also might move into this bad environment where maybe you'll produce fewer individuals, but again, you can maybe exchange back. So actually, this is very similar to kind of more classic ecological models where we have kind of source sink dynamics ongoing. So we want to then think about, okay, how do we model this process? So again, we've had some nice introduction kind of yesterday from Jeff about kind of class reproductive value. So what I'm going to do is we're going to try and understand how genes are flowing between these states by defining this matrix P. And so we're going to say that we can move back in the population through time. So with probability A alpha, we're going to stay in the same state, and then probability 1 alpha will move between states. And the same for our kind of mutation linked to our deleterious copy. So the kind of top row is if we're linked to our beneficial copy, and the bottom is if we're linked to the deleterious copy. And so what we can do is we can calculate the kind of stationary distribution of this matrix, so kind of the long-term amount of time we spend in these two states. And so that's analogous also to thinking about how much are these two states now contributing to the long-term future of the population. So there's that nice kind of back and forth analogy. And so then we get out these results. And then the other part of this is why are these quantities useful when we're thinking about selection? Well, we can think about a kind of perturbation to this population. So we can imagine if we're one of these gene copies upstream and we want to do something slightly different, our dynamics are going to be different from the mutant. And so we can consider, will this mutant increase or decrease in frequency? And so normally we'd calculate like an eigenvalue. So it's really an eigenvalue problem. But maybe to make that simpler, we can write that eigenvalue as a kind of series in a perturbation. So the nice thing is actually just if we think about the first term in that perturbation series, we get this really nice result where it's just we need to weight the kind of selective effects moving through our life cycle. So like SG and SB are my stupid namings for like the good and the bad gene. So we can imagine that these might be positive or negative, but this first term is just going to be, we're going to weight these selective effects by how much time we're spending in these two states on average. And so we can write this out in kind of like a matrix form, but for this to invade, we need this to be greater than 1, greater than 0. We need this to be positive. So then coming back to this recombination and linkage thing, if we imagine we're a gene and we're kind of sitting at kind of various degrees upstream of this deleterious locus, we can write out our matrix as I described before. And so with some approximations, we can write it out in this way. So if you think about you're on the good copy, what's the way that you might not be on the good copy in the next generation? Well, it's going to be roughly the probability that you mate with a bad copy, and then there's recombinations. That's going to be the kind of main process that's going to exchange you between good and bad copies if you're currently on the good copy. And then if you're on the bad copy, we're going to imagine that a bunch of them die. That's the S. And they're going to have to be replaced by the good copies through maybe like a mutational process. So we're going to there's going to be kind of some killed and some replaced. And so that's going to that's going to give you new copies coming in. And similarly, there might also be recombination, but we can imagine that if you're a bad copy, basically you're always mating with good copies because you're much rarer in the population. So that's before we can calculate the reproductive values for these different states. And then the interesting thing is we can think about, okay, if we're different degrees of linkage to this deleterious copy, what is our kind of reproductive value distribution going to look like? And again, everyone's going to have the same frequency of the good and bad copy in total in the population. We're just kind of moving along different degrees from this focal bad copy. So what you can see is I've just written this as like the individual reproductive values. That's just the class reproductive value divided by the total number of individuals in that class. So the main thing I want you to take from these graphs is if we're perfectly linked, so there's zero recombination, those that are on the bad copy are going to be worth nothing because you're going to die eventually. You're linked to that deleterious copy and that's not going to contribute to the future. And then as we get further away, you've got a greater chance of being able to swap onto those good copies. And so actually, you're slightly more valuable in the population. So we can see that actually, recombination really matters here in actually deciding if I'm a gene and how much do I value maybe improving the bad copy, and maybe this is at some cost to the good copy. I have to make some trade-off on, maybe this would be like mutational robustness or something like that. Actually, the value that we're going to balance between the good and the bad state is going to vary depending on our recombination rate and how far we are away from this locus. So we can see that this has the potential then to generate intragenomic conflict between different regions based on how far or closely linked they are to deleterious copies. So I've kind of just summarized that logic here, where this differential linkage to loci which have fitness effects is going to generate asymmetries in the reproductive value of different copies in the population. So I'm not the first person to come up with this idea, obviously, but there's a nice paper where they look at not deleterious mutations, but instead they look at local adaptation and plasticity. And so you see a very similar logic in this paper. So it doesn't necessarily have to be kind of straightforward mutation selection balance. It could be many other processes that have copies which have differential fitness in different environments. So that's the first thing. So these conflicts, how much time do I have? These conflicts only refer to this first order term. So as I said, there's lots of other orders in this perturbation that might really matter, because we can imagine, actually, if we do something, this is going to alter what state we're in the next generation. Actually, that can really matter if selection's stronger or how we move between the states is slightly slower. And so we have to try and think about how do we account for the fact of how selection maybe perturbs how we're flowing through the life cycle. And then this can then feed back onto the invasion conditions. You can imagine if you move into the good state, then you might spend more time in the good state as a kind of mutant lineage. So how do we calculate that? So so this is kind of an approach that we can use. So first of all, we have to define another matrix, and this is what's called the deviation matrix. And this matrix, maybe this doesn't look like it means much, but the idea is that each of the entries of the matrix is asking, if I'm in one state now, so say I'm in a male or a female, how much longer on average, how much extra time on average am I spending in males and females? So this is kind of how we calculate it, but maybe to make it more concrete, let's think about an X chromosome. And imagine I start off an X chromosome system and I start off in a male. So that's kind of here. So I'm starting off in a male. I'm definitely in a male. I'm definitely not in a female. So you can imagine generation one, kind of this, and then I calculate, oh, this is the long-term frequency I'd spend in this state. So it's kind of, I spend a little bit more time in a male, less time in a male, and we sum up all these differences from the stationary distribution, and then we get out these quantities. And so our matrix is just populated by those different entries. Or alternatively, thought about an autosome, it's just going to be minus a half and a half, because actually what's nice about these genes is they immediately attain the, kind of after one generation, they're immediately at the stationary distribution of how much time on average they're going to spend in males and females. So there's this nice property where actually when we think about the higher order terms in our perturbation series, it turns out they're related to this matrix. And that kind of makes sense, doesn't it? If we think about selection in one state is going to have some perturbation, maybe increase or decrease our frequency in the other states. And then we're going to think about, oh, actually, we have to kind of run that through of, well, okay, how much extra time am I going to spend in a male or a female? And that's what our matrix, our deviation matrix I've just described is doing. So this is kind of our second order term, but actually the same logic kind of constantly occurs for the higher order terms, and they're just more and more complicated versions. And actually we can just sum this up and get the kind of exact invasion condition as written in this way, where it's really just a combination of the kind of neutral reproductive values, the kind of perturbation we're doing, so kind of selection coefficients, and then this deviation matrix. So kind of this is written out in matrix form, but yeah, the kind of that's the core logic. And then so one thing I've trying to work out is what's the best way to describe this. One way is that you could think about an altered class distribution, our mutant experiences, to try and combine them all into one set of terms. Another way we can do it is to think about, we can decompose it and think about, this is our kind of standard first order approximation that often we use. So where we just think about selection weighted by these neutral reproductive values. And that's exact if we imagine that selection only happens periodically. Say our mutant only expresses its deviant phenotype every, 100 generations, then what this is doing, this will go to 0, because this is really telling us how the combination of selection and how we move through the life cycle are kind of compounding one another. So this is kind of describing our mutant kind of alters its distribution, and then kind of the population will mix and relax that, but it's that combination of kind of perturbation and relaxation that's kind of giving rise to this kind of distortion term. And so I think this is actually basically the same as like in a kind of QLE analysis people do. These are kind of disequilibrium terms where we're thinking about how much or how little you kind of overexposed in one lineage or another. So to make these ideas more concrete, we can think about a nice biological example of sexual antagonistic pleiotropy in the pseudo-autosomal region. So again, we can think about, we've had some sex chromosome introduction. So you can think about this is like an X chromosome system. So our females have 2 copies and our males have 1 X chromosome, and this is the kind of Y chromosome. So we think this is our sex determining locus. And then this is maybe the region we're tracking upstream of this. You can think about maybe our region is going to balance, it might have some good effect in a female if it does one thing, it might have some good effect in a male if it does the other, but it has to trade them off. So again, we can describe, as we did for the kind of mutation selection balance case, you can think about how we're going to move between these different states. And it's going to be a combination, it's going to be determined by how often we're going to recombine away from this sex determining region. So we get kind of our matrix looks like that. And so the nice thing is this example is identical to the autosomes in actually how much time in the long run we're spending in males and females. We're spending half the time in the female and a quarter of the time on the X chromosome in a male and a quarter of the time on the Y chromosome in a male. So this is essentially half and half in males and females, identical to the autosomes, but actually how it's moving through the life cycle is different, and it can be different depending on what that recombination rate is. So then we can calculate the invasion conditions for maybe one of a sexually antagonistic allele. So on the kind of X-axis, we have the kind of effect in females, and on the Y-axis, we have the effect in males. So basically, below these lines, the kind of female beneficial strategy is going to be invade, and above these lines, the male beneficial strategy is going to be able to invade. And the region between the two is a kind of polymorphic region. So the kind of solid lines here I have for the kind of classic autosomes. So this is nice because this is both the autosomes, but it's also that first order term, because this is the autosomes in this pseudo-autosomal region have an identical first order term. The only thing that then differs is how these kind of distortions are building up on the pseudo-autosomal region versus the rest of the genome. So we're going to see that actually on these pseudo-autosomal regions, depending on the recombination rate, if it's very low or a little bit higher, we can more easily get kind of, in this case, it'd be kind of male beneficial, female costly alleles. And here we get an extra little bit of space for kind of female beneficial and male costly alleles. But the important thing I want you to take from this is the kind of long-term valuation of males and females is identical for these regions of the genome. The thing that differs is how this distortion builds up. So we can see there seems to be a form of intragenomic conflict that's arising from this combination of how selection and the movement through the life cycle is building up as we move along our genome. Okay, so do I have, what's the? Minus 2 minutes. Okay, well, I'll quickly wrap up. Okay, so the idea, the main thing I want to take from this is actually linkage can be really important both in shaping the kind of valuation of different states, but also how these distortions build up and how genes might actually, a mutant lineage, spend more or less time in particular states. And the reason I think it's interesting is actually coming back to this paper is that the kind of same kind of ingredients that kind of can generate these conflicts actually appear to maybe in a lot of other modifier models. Many cases you'll see recombination is important for the invasion of a strategy. So it might be that, in many cases conflict is only transient in these cases where maybe it's only when the mutant's invading, and once they've kind of got this new strategy, everything reached a nice equilibrium, you don't want to go back to what you did before. But it could be there are also cases like this enhancer runaway, where if there's fixed costs or differential use of resources, then this might generate ongoing conflicts. So yeah, okay, sorry. Yeah, that's everything I